Retinitis pigmentosa is an inherited degenerative eye disease that causes severe vision impairment and blindness. Expression of RP is inconsistent. Some individuals may exhibit symptoms from infancy, while others may not express this until later in life. RP is caused by abnormalities of either the photoreceptors or the retinal pigment epithelium of the retina, which leads to progressive sight loss. Symptoms such as defective light-to-dark or dark-to-light adaptation and degeneration of peripheral vision may be expressed by affected individuals. There are multiple genetic mutations that can cause the RP phenotype. Mutations in the rhodopsin gene are known to cause approximately 25% of autosomal dominant forms of RP. In wild-type genes, rhodopsin proteins are G-coupled receptors involved in phototransduction. They are highly sensitive to light and drive a cascade of biochemical events which enable vision in low light conditions. Mutations in rhodopsin genes may induce changes in the amino acid sequence. The primary mutation in rhodopsin gene occurs at codon 23, in which a proline is changed into a histidine. This induces the rhodopsin protein to misfold, leading to photoreceptor degradation, which ultimately results in severe vision impairment.
In our experiments, we will use the model organism, the house mouse, to investigate the effects of mutated Pro23H in the orthologous rhodopsin genes in retinitis pigmentosa. Since histidine is a basic amino acid residue, we want to determine if this misfolding is based on the charge of the amino acid substitution. Thus, we will use a substitution from proline to another basic amino acid, lysine, coded Pro23K. First, we create a homologous recombination construct using a mutant allele from an affected individual, a missense mutation Pro23K, GFP neomycin resistant selectable marker, and flanking rhodopsin gene sequences for a targeted mutation. Second, we inject the construct into embryonic stem cells, isolated from a white mouse blastocyst and grown on a petri dish. Cells with the construct will be selected for using a neomycin culture. Cells without the construct will not survive due to a lack of neomycin resistance. Selected cells will be removed from the petri dish and injected into a gray mouse blastocyst that will be implanted in a gray mother mouse. The progeny will display patches of gray and white, indicating presence of wild-type and recombinant cells. Backcrossing the chimeric progeny with a gray parental strain will yield impure recombinant mice, which we can then screen for the RP syndrome. If the recombinant mice display RP, then we can conclude that Pro23K mutation has a similar effect as a Pro23H, and that protein misfolding in RP may be a result of a change of amino acid charge.